Good morning. I will present the initial results on the effect of GAP27 on the development of breast cancer brain metastasis. So, the interaction between tumor cells and the microenvironment is crucial in the development of tumors and metastasis. Recent evidence suggests the role for transmembrane connexin proteins in these tumor cell-host cell interactions, as well as in extravasation and survival of metastatic tumor cells. Transmembrane connexin proteins are the main component of gap junctions. As you can see in the figure, 6 connexins form a connexon or a hemichannel, and the docking of 2 connexons leads to the formation of a gap junction. A gap junction is an intercellular channel which allows small molecules and ions to pass through from one cell to another. Connexin 43 is expressed by the brain endothelium and has been shown to have an increased expression in metastatic breast cancer cells and metastatic melanoma cells. In the formation of metastasis and increased connexin-intermediated intercellular communication is shown between the metastatic tumor cells and the endothelial cells, and also between the metastatic tumor cells and the astrocytes. The gap junctions between the endothelial cells and the metastatic tumor cells will allow the metastatic tumor cells to grow into full-blown metastasis. The gap junctions between the metastatic tumor cells and the astrocytes provide protection of the tumor cells against drug-induced apoptosis. In a study done by Salatif and colleagues, the role of connexin 43 in mediating the early events of brain colonization was investigated. In the first study on the left, the 41 breast cancer cell line or the 41 breast cancer cell line with the connexin 43 knockout were injected in the bloodstream of chicken embryos. After 7 days, they evaluated the brain colonization, and there was a significant decrease of brain colonization by the metastatic tumor cells displayed in green in the brain vasculature of the chicken. So they could say that metastatic breast cancer cells require connexin 43 for extravasation and brain colonization. In the study on the right, they looked at the effect of the gap junction inhibitor carbonoxylene, and they also saw after administration a decrease in the brain colonization when the gap junction communication was blocked, which also leads to the conclusion that connexin 43 or gap junctional communication is necessary for brain colonization and microtumor formation. The gap junctional communication can also be blocked by GAP27. GAP27 is a connexin mimetic peptide and is identical to a sequence on the second extracellular loop of connexin 43. The exact working mechanism of GAP27 is so far unknown. So we wanted to investigate the effect of GAP27 on the development of brain metastasis in female nude rats. We used the MDAMB 231BR breast cancer cell line and labeled these breast cancer cells with micron-sized particles of iron oxide. Then we injected 2 groups of female nude rats, a control group and a treatment group, with 100,000 of these cancer-labeled cells into the left ventricle of the mice using ultrasound guidance to target the left ventricle. The treatment group received, together with the labeled cancer cells, a co-injection of GAP27, and the following 2 days after the intracardiac injection, the treatment group also received intravenous injections of GAP27. The control group received at the same time point PBS instead of GAP27. The day after the intracardiac injection, we performed a high-resolution MRI, and you can see here, what you can see here is the T2 star weighted images that shows the initial cell distribution and cell load. So the black dots are the cancer cells labeled with micron-sized particles of iron oxide. The weeks following the intracardiac injection, we also performed high-resolution MRI, and you can see here T2 weighted images. So the first signs of metastatic colonization were visible as hyperintensities on these two T2 weighted images at week 3. At week 6, our experiment ended, and then we performed histology to correlate with the MR imaging. The histology is the hematoxylin and eosin staining of established metastasis in the cerebrum. And you can see on the left normal brain tissue, and on the right you can see the metastatic tumor in the brain. So our results, evaluation of the number of metastases at week 3, 4, and 6 did not show any significant results. But when we looked at the volume of the metastasis, we saw a big difference between the control group and the treatment group at week 6. So the volume of metastasis was significant between the two groups. To conclude, early administration of GAP27 has no significant impact on the metastatic seeding in the brain, but we saw a reduction of metastatic colonization in the brain. In the future, we will do some further experiments with GAP27. We will continuously administer GAP27, and we will also treat the established metastasis starting at week 3 with GAP27. We will also do experiment with a connexin 43 antibody in the future, and we will start again with the early administration like we did in these experiments I've just shown. Thank you for your attention.
My intelligence is probably deteriorating, but when you say injecting gap junction, could you explain what that means?
Excuse me?
You're treating those animals with the supposedly gap junction protein.
Yes.
What do you expect? How do you expect that to be integrated into the formation of gap junctions? And for example, with connexin 43 being one of the major components of gap junctions, if you want, first of all, by the tumor themselves get reduced expression of those gap junction proteins, but if you want to induce an increase in the number of those gap junctions, you need to transduce the cells to produce the protein. So I'm not sure exactly what you did. If you could briefly elaborate on how you changed, modified the expression of the gap junction in the animals.
We just administered GAP27, which is a gap junction blocking peptide. It blocks connexin 43, which is a component of the gap junctions. So the communication between the endothelial cells and the tumor cells can be stopped, and the growth of the tumors can be stopped or can decrease. That was our main goal. So.
Okay. So it's an irreversible blockage of the gap junctions that you're trying to achieve?
Yes, but we don't know the mechanism of GAP27 so far. That's something we need to find out, and we will do this with in vitro models in the future.
Do you have any electron microscopy corroboration of this phenomenon? Because some of those movements across gap junctions can actually be seen.
No, not yet.
No. Any questions from the audience?
Yes. Are you planning to label this inhibitor to see where it accumulates in those metastases that are established? That it seems that the inhibitor is very effective in blocking the outgrowth, but maybe not the very first steps. So do you have any reagent that allows you to label this molecule when it binds to the connexin, to analyze how is the biology behind?
Yes, that is something we will do in the future. We will label GAP27, but it's for in the future, so we haven't did that yet. But we are planning to do it.
My question goes into the same direction. So, so your data with the same number of mets, but just very over time decreasing size just speaks for a treatment effect, right? Not a prevent effect. So these endothelial cancer cell interactions probably are not so important in this system, but interactions between cancer cells or with other cell types in the brain. So do you have an idea what is the most important? I mean, because your hypothesis that endothelial cell might not really be the cell type that is of importance here.
So far we are not sure where GAP27 exactly works. It could be that it works between the tumor cells also, but there are things that we need to figure out in the future.